So the major task for doctor is to perform correct diagnosis, because to avoid the disease, we should know about the disease. If you know that subject is infected with HIV infection, you will follow precaution measures. So how to perform diagnosis of HIV infection? Of course, diagnosis is complex and involves several different tests. We have multiple serological tests, such as ELISA. We have nowadays immunochromatography tests, rapid tests, and they can be targeted to identify antibodies to virus or antigen of the virus, depends on the stage and type of the test. So first thing you should remember that we have a window period. It is time period when subject is already infected with the virus, but virus is not multiplying actively. It is the stage of adaptation, and antibodies are not produced yet. It is usually about a month or 28 days between episode of infection and first appearance of virus in blood and in tissues. In first 28 days after infection, any test will show you negative result, but subject is already infected, just virus is not active. Second thing, no test performed in single manner can provide a diagnosis of HIV infection. To put the diagnosis of HIV positive, you need to perform as minimum 2 different tests performed in time interval, and in some cases, in some countries, it's even 3 tests should give you positive result. And first 2 tests are usually screening tests available in any lab, and to confirm the diagnosis, we always need to perform immunoglobulin analysis. Only that test is confirmatory for HIV positive state. We can talk again a lot about prevention. If you know measures to avoid sexually transmitted diseases, you will know what to do here as well. Of course, it is use of barrier contraception during sexual contact. Medical staff should use common measures such as gloves, if required, protective glasses, or use of antiseptics. All instruments should be single use for sterilizing correct manner. Of course, any activity with samples with biological liquids, with the blood or other, should be performed only in specialized laboratories with, again, gloves to avoid any contact. For medical surgical staff, we have specific gloves. With person who is confirmed with HIV infection, especially if it is a medical staff. Again, you have post-exposure course of antiretroviral therapy, which should be initiated within first 72 hours after exposure. It is important when subject is delivered to medical institute or some hospital in emergency state. You don't have a time to diagnose the disease, to perform any test. You need to give a medical help right now, and during those emergencies, very often accidental contact with the blood or some punctures with bones or other sharp instruments can occur, and doctor will be exposed to the blood or other biological liquids of the subject. And after that, when you perform the test, it will become known that subject is HIV positive. Doctor should start preventive course of antiretroviral therapy as minimum for 28 days. Of course, in those cases, medical institution or hospital should pay for that course, or government will pay for that course. It's not for the doctor. And of course, that measure can help to avoid and to prevent infection with HIV virus in people, and virus will not invade and will not infect the subject. If pregnant women are infected with HIV infection in kid delivered healthy, or maybe woman became infected after birth delivery, then of course it's better to avoid breastfeeding or give antiretroviral drugs for pregnant women to avoid the disease in fetus, and for breastfeeding women to avoid transmission of virus to kid. If you talk about therapy, we do have therapy, but actually that therapy should be first lifelong. Second, it should consist of 3 or more drugs, which should be regularly switched. And of course, that therapy can only suppress replication of virus and prevent progression to AIDS state, but infection will remain lifelong. We still don't have any cure for the disease. We cannot remove virus from human body. And actually, officially, again, it is a huge company performed by World Health Organization to provide free treatment for infected people worldwide. And according to official data, in the last centuries, number of deaths due to HIV infection decreased almost twice by 51%. And in that time period, almost even more than 15 million people were saved from death or from complications of human immunodeficiency infection. If you talk about treatment, here in the slide only main drugs are presented. We have multiple different groups of therapy because of serious impact and significance of that infection. Of course, a lot of companies are trying to design some drugs, and we have a lot of options. Drugs of different groups can act on different stages of viral replication or life cycle. And as you can see, a lot of available here. So nowadays we have already combined drugs which are produced, contain several active agents in one tablet. And of course, because some of the drugs are registered far ago, we have generic therapy, generic options when cost of drugs are much lower than original drugs. And nowadays, actually, it is possible, and a lot of people can afford that therapy, even if it is lifelong, to prevent progression to AIDS syndrome. So the major, again, option is to identify disease in a timely manner, and we have several tests. The most popular nowadays screening test, it is immunochromatography. As you can see, it is a common test as it was shown for other infections, cartridge or plastic device, which is used to add samples and to indicate the result. Paper strip or some synthetic material strip which contain required indicators for the disease, and you need to add a sample of blood into the corresponding window. It will fill in that paper strip, add some reagents which are going in the kit, and in the window, result window, you will have control strip and a test strip. So in modern tests, we have we have an option not just to see positive or negative result. We can evaluate even activity or load of virus or load of antibodies by changing of color and of that strip, or you see that it is a dark blue and pale blue, or you can measure the zone of that result. Larger zone indicates more amount of antigen or antibody present in the subject level, so we can approximately evaluate activity of the disease and the stage of the disease. Second option, of course, it is classic ELISA test performed in microarrays. Again, and nowadays usually they are combined. They can identify both first and second type of infection. They can identify antibodies or antigens. That test again is screening, and it is essential to obtain positive or negative result. In some cases, we have differential systems, the same ELISA, but contains several different enzymes, which will give you different colors for HIV-1 or HIV-2 infection, or intensivity of color in the well. In the left picture, you can see that almost all samples are dark brown, but one is pale brown or yellowish. It means that few only antibodies or antigens is present in the sample, and intense signals in the lowest wells will indicate that antigen is present in high amount or antibodies produced in much higher amount in other cases. So again, we can approximately evaluate microbial load or intensivity of immune response. Western blot analysis, again, we discussed it during precipitation test. It is a type of immunodiffusion in gel. In that case, you will need to take patient serum to identify antibodies against precise known antigens and add that antigen in the gel. If antibody will match antigen, they will form zone of precipitation. It is easy seen by formation of those black lines. And you know the place where antigen was added or molecular weight of those antigens, and formation of that zone of precipitation in known area will indicate presence of antibody against known antigen. So the test is confirmatory because other tests cannot differentiate between antibodies and antigens, but here you will see precise antigen which inducing immune response against it. And usually the test is evaluated as positive, strong positive, or weak positive, and negative. If only few, one or two antigens, antibodies against few antigens identified, it is a weak result, weak positive. But if three and more antibodies against antigens are present, it means that result is for sure positive. And again, by evaluation on different antigens, you can see which type of virus is present in subject and how strong is immune response, approximately again which stage of disease is present in those person. So that symbol is one of the most known again in nowadays. It's red strip and that shape. It indicates, and usually it is present in the clothes of some people. They can wear it in different parts of the clothes. If you see that some person wearing that symbol, usually it indicates that subject can help you to obtain correct knowledge about disease. If you have some problems with relatives or friends, you can ask where to look for medical help, and usually they can give you some contacts of institutions who provide free diagnosis and anonymous diagnosis and free treatment of the disease. So if you have any doubts, any problems, you need help, you will see that symbol. You can absolutely free ask that person what to do, where to seek for the help. They will not share that information with anyone. They will not make doubt for you. They will just give you contact information or will give you consultation, which is absolutely free, and they will try to help you.